So this will be the end of the optimization module, okay? Any questions so far? Okay, now I'm going to start the next module using a different problem to motivate this formulation. Okay, so we're talking about machine learning, but you know optimization and machine learning are two sides of a coin. Okay, so I'm going to start with the protein function prediction. So far we're doing sequence and structure, but don't forget function. Okay. So where we are right here. Okay, we're going to use this to explain the basic concepts, but I think many of you already know machine learning already, so I'm going to go through this part really quickly, and sometimes I will just directly ask you, what do you recollect for those concepts, okay? At the end of today's lecture, I will ask you a question about a support vector machine, so please don't let me forget about it. Please remind me, okay? Okay, so motivation, right? So we're now moving to the next question, right? Protein function prediction. So why, what, and how? Okay, and then how this motivate, how the challenge motivated machine learning models, okay? So why? We know that there is a gap between the sequentially determined and the structurally determined proteins, right? But also the gap between the sequence determined and the structurally annotated, functionally annotated proteins are also wide, okay? For instance, this is the green one, Swiss prot. Okay, this is the manually annotated protein sequences. Okay, those sequences are already manually annotated with the function. Okay, but there are way more sequences, right, in Tremble, which include a lot of sequences that haven't been annotated or just annotated automatically. Okay, so these three data sets are getting bigger and bigger. Protein sequences that have been structurally determined, protein sequences that have been functionally annotated, and protein sequences that have been collected. Okay. So, and if you look at this genome-wide protein function prediction, this is old data, but still more than 40% of the known sequences, right, in our species. Even after you exhaust the homology-based method for annotation, you still end up having like 40%, okay? 40% of the molecular functions unknown. So now the question is, how do we do that? Again, why is this a computational problem? You told me that sequence is 1D, structure is 3D, I understand. But how do you even describe a function? I don't even know what's the dimension, right? So like what we did before, we looked at the sequence structure relationship before and ended up knowing that there are some heuristic rules. Then we can look at the 30% as a cutoff, right, and figure out whether we are in the safe zone or not, right? Here I'm going to do something similar, but we're going to talk about more about it. So in general, there's no golden rule, okay, but there are some heuristics, like we said before. Safe zone is 30%, okay? And we knew that the structure is more conserved than sequence, right? And this can be explained by these bullet points that we have learned before, okay? And it's like the 1 versus 100, okay? Then what about the sequence versus function? Well, the safe zone is now at about 40%. If the identity is about 40%, then the sequences tend to have similar structures and functions, okay? But again, it's a heuristic. What about the structure versus function? Well, it's more complicated and less well known. But because one issue is that I cannot say that similar structures give you similar functions because I haven't described the functions yet. One structure can have multiple functions. Right? So this is another issue. And also your data might be biased because, look, all the protein sequences that have been structurally determined are already biased those structures are structures that the current technology, like NMR, X-ray, can collect, right? So that data set is already biased. So I cannot really derive my conclusion from such small data set, right? So let me give you one example. Okay, so this is one structure, multiple functions, right? So this protein is an enzyme. It's called glucose 6-phosphate isomerase. So when you see an ASE at the end, you know that it's kind of an enzyme that makes some chemical reaction much faster. And this enzyme is basically converting the glucose 6-phosphate substrate to fructose 6-phosphate, which is a product. This is what this enzyme do, but it's just one function, okay? So if it's forming a dimer, this is a dimer, okay? You have this one monomer in the front, one monomer in the back, right? It's symmetric. So this dimer in the cell is accelerating this chemical reaction, okay? But it could be a monomer outside of the cell, right? Which act as neuroleukins, cytokines, and so on. So they could have different functions, okay? So now you see there's a problem, right? Functions, first of all, there's no natural dimensionalities to begin with. Secondly, function is not just sequence and structure determined. It's also context dependent, right? If it's in the cell or it's outside of the cell, if it's a dimer versus a monomer, or right? Or, you know, the function could be what partners or protein interactions you form, right? So that's a complication. So then how you define functions? The way you define protein functions, how many of you have heard of this gene ontology? Good. So it's okay, I'm just trying to figure out. So gene ontology is basically consisting of three ontologies: molecular function, biological process, and cellular component. Okay? Molecular function says, what does this protein do? Biological processes ask, what processes is it involved in? And the cellular component asks, where does that happen? Okay, so this is why we have three ontologies. Okay, the reason that we call it gene ontologies is because proteins are gene products, right? Remember, it's translated. So if you need to know what ontology means, we have a lot of ontologies. It's a formal representation of a body of knowledge within a given domain. Okay, so there are a lot of, it's describing a lot of relationships between classes or terms. Okay, here we have three ontologies, right? Okay, as I said, cellular component, biological process, and the molecular functions. Okay, this is one of the earlier versions, but you can go to geneontology.org and you can find out that in each ontology we have certain number of nodes, and each node is one specific molecular function, for instance, and each node may correspond to multiple proteins. In other words, multiple proteins may all reside or share that function node. Okay, so for instance, right, this is a simple illustration. We focus on the biological process rather than these other two. Okay, let's start with the biological process. Then we basically see that there are a lot of nodes and a lot of arcs or edges, right? So there are actually many different kind of relationships. But in general, as you start from, now this is like an upside down tree. Okay, if we only consider certain relationships. So on the top you have the root. As you go down to the leaf, you see that if you follow a path, the function is getting more and more specific, isn't it? Isn't it? For instance, here is a regulation of biological process. This is a positive regulation, right? And for in this case, pigmentation, and this is the pigmentation during development, and so on, right? So it's getting more and more specific. But it's not general because it depends on what kind of edges you consider, okay? I means is a, so it's like an instance, right? So in this case, pigmentation is a biological process. Pigmentation during development is a pigmentation. So I means is a, but there are other kind of relationships, okay? If you only consider is a, this relationship, then this graph can be roughly regarded as a DAG, a directed acyclic graph. But if you consider more than this simplified ontology, then this is no longer a DAG anymore, okay? So what we often do is a simplified graph, but it also has limited information, especially the relationship we can have here. Any questions so far? So, how can you predict a protein function, right? What are the inputs you can do, right? I'm not talking about how you represent the input. I'm just asking what kind of input, right? Apparently we said that there's a sequence, there's structure, right? How do we represent the sequence? How do we represent the structure? That's a different issue. Like, you observed in the threading how we represent the query sequence or how we represent the template structure, right? We could use profiles, we could use the secondary structures and so on. That's how you represent, right? But if you ask the type of data, we have the sequence, we have the structure, and there's more than that, okay? For instance, you can look at the protein-protein interaction networks, okay? So if you ever, met some new friend and you want to know more about him or her, right? But his Instagram is not open to everyone, right? But then you find a mutual friend, maybe. Then you guess, well, maybe they share similar interests, right? Right? So in other words, you may not know a person well, but if you know that person's companions, then you can probably tell what that person is doing. So this is a Jewish proverb, okay? Tell me who your companions are, and I will tell you what you are, okay? Another kind of data is the protein expression profiles. So in different environments, you have different genes or proteins of different expressions, okay? Sometimes you may see that some genes or proteins expressions are having a strong correlation. They both go up or down together, or one goes up, one goes down. We'll continue the introductory of the second module, machine learning, and we are motivating this topic with the problem of protein function prediction, okay? So we talked about what is a protein function, or more specifically, how can we describe a protein function? Can someone tell me how we describe a protein function? Using what? 2 words. How do you describe a protein function? Gene ontology, GO terms, okay? There are 3 ontologies: molecular function, biological processes, and cellular component, okay? So it's about what these individuals do, what is it part of, and where does that happen. So these are the ontologies, okay? All right, so now let's, think about this. You learn a lot of machine learning courses already. So here I'm just motivating the fact that we are using a lot of data and integrating them together for the purpose of protein function prediction, right? So I'm giving you one example, right? So here, you can jointly embed the input features and the output labels. So the input is a sequence which is in a text. And the output is defined as probability over the gene ontology, which is a graph, right? So in this sense, what you can do is that you can jointly embed the input sequence and the gene ontology labels, right? So that after embedding, right, they're sort of close to each other with some proximity. And then with this learned embedding, you can do this prediction using fully connected layers for the probabilities of the geo terms, right? So this is just for individual proteins functions. But you understand that this is just one kind of signal or input, protein sequence, right? There are a lot of other data, like what we described before, including protein-protein interaction networks. If you think about it, actually, if you only rely on protein sequence to make a prediction on the molecular function, maybe that makes sense to some extent. But if you think about the second ontology, which is biological process, right? It's asking who are you teamed together with, right? So in what big process are you part of? So in that case, you definitely want to use additional data, right? Because this protein-protein interaction network must tell you a lot of information about the biological process, right? So, and then there's a cellular component as well. So in other words, in general, you would imagine that if you use more data that are more related to the the gene ontologies, you may get better results, and you have a rationale about that, right? Especially because of the other ontologies, other than molecular function, are encoded in more than protein sequences. It's also encoded in the protein interactions and so on, right? And expressions. So a quick question for you is that now, okay, here I'm embedding the individual protein sequences and a constant label matrix, okay? How do I incorporate the protein-protein interaction data into this framework? Any ideas? Again, here I'm embedding the individual protein sequences as text, right? The additional trick I used is to embed the graph so that the embedding of the sequences and the embedding of the graph labels are kind of close, right? In the latent space, right? You can forget about this for the moment, right? Because this is a constant branch for an arbitrary protein sequence, right? Because the label is unknown. This is a fixed K by K matrix where K is a number of geo terms on the gene ontologies, right? So if now you want to incorporate additional data about protein-protein interactions, what would you do? Any suggestions? How would you model protein-protein interaction data? What kind of data modality is this? And what kind of models are good at learning from such data? Well, you can represent the protein-protein interaction network as a graph, right? And what can you use to model graph data, for instance? There are many choices, right? For instance, I can pick one of the latest hot topic, graph neural networks, right? So then on this graph neural network, right, what is each node? What does each node represent? The graph is for protein-protein interaction. So what is each node? A protein, right? You follow? So this individual protein encoder, right, used to be IID in the training set, can now be correlated with each other because on each node, you have such a sequence embedder the same sequence embedder, but with different sequence inputs. Is that right? Right? So in other words, you are training two things together. One is how to embed the sequences. Two is how these interacting proteins affect each other's embeddings, right? You're learning the text embedding and the graph embedding together, right? So that's one example. Does that make sense to you? Okay, this could be a final project, okay? Any questions? Okay. So let's again try to look at this problem. It's actually done in a community assessment called CAFA, Critical Assessment of Function Annotation. There was an old summary which says that the challenge for these methods, first of all, you're looking at half of the methods use data beyond the sequence similarity. The sequence similarity is the basic thing, right? So this joint embedding we did was just digging the sequence similarity in the latent space, right? But still, there are evolutionary relationships, protein structures, protein interactions, and gene expression data, okay? So the challenge for these methods was finding ways to integrate disparate data sources and properly handle incomplete and noisy data. Like what I said, right? You cannot just featureize them and concatenate them and just dump them into a generic neural networks, right? So most methods on the computational side use a machine learning principle, okay? So, but they do require experiences in the classification models, learning parameters, feature of the training set, and so on, right? And among the three categories, performance may be considered accurate for molecular function, which is mostly determined by protein sequences, but in the case of a biological process, the overall performance was below, okay? Okay, so we actually have the CAFA test set, right, accompanied with this paper. So we can compare our performances to the whole community as well. So in this sense, let's now think about it. Why, right? Everybody is using machine learning to solve this problem for a reason, right? Not because machine learning is hard, right?